I'm Csaba Orban. I'm a senior research scientist from Dr. Thomas Yo's lab at the National University of Singapore. I'm going to talk about how irregular heart rhythms can help us dissociate cardiac modulation of the fMRI signal from the effects of respiration. I have no disclosures to declare. When a mouse's whiskers are touched, neurons in its barrel cortex are depolarized, which causes nearby arteries to dilate. This leads to an increase in local oxygenated blood flow, peaking approximately 5 to 6 seconds after the onset of neural activity. This is the natural phenomenon fMRI exploits to help us infer the spatiotemporal structure of brain activity. In addition to local neuronal activity, the heart and the lungs can strongly influence blood flow and therefore impact fMRI measurements. Studies looking at cardiac and respiratory effects on fMRI have revealed highly overlapping temporal, spatial, and spectral effects. Identifying the unique contribution of cardiac rhythm and respiration has therefore been complicated by their intimate interactions. Much of cardiac dynamics is coupled to respiration. Sinus arrhythmia involves the entrainment of heart rate to the phase of breathing. Despite its name, it's a feature of a healthy heart rhythm. Deep breaths and sighs occur spontaneously to help inflate collapsed alveoli, and these typically cause excursions of heart rate, as recently shown by Jonathan Power and colleagues. Transient arousals that arise in the brain during sleep can also trigger changes in heart rate, pulse amplitude, and respiration, as shown by Pinar Osbe and colleagues. Finally, afferent nerves may also transmit sensory and interoceptive signals that arise due to respiration or cardiac rhythm to the brain, which in turn may influence the fMRI signal via neurovascular coupling. One way to isolate the effects of the heart could be to study arrhythmias. Healthy heart rhythms are initiated at the SA node, which sends impulses down a conduction pathway, ensuring the timely contractions of the atria and the ventricles. During a premature ventricular complex, the ventricles depolarize spontaneously, producing a short coupling interval followed by a long post-ectopic interval. This is a common occurrence even in healthy individuals, which is sometimes experienced as a skipped heartbeat. This generates a transient drop in blood pressure due to the fast premature beat not leaving enough time for the heart to fill with blood, and due to the extended post-ectopic pause. The drop in blood pressure engages a baroreflex, which generates an overshoot in heart rate and blood pressure. Since PVCs are triggered at the level of the heart, they may allow us to observe cardiac modulation of the fMRI signal in the absence of respiratory influences. We explored this in resting state fMRI data from the Human Connectome Project, or HCP, which has around 1,000 participants scanned for almost an hour at a high temporal resolution with concurrent acquisition of pulse oximetry and respiratory bellows signals. Unfortunately, the HCP did not acquire ECG data, which is the gold standard for studying heart rhythms. However, we could still explore the heart rhythm based on heartbeat intervals defined by pulse oximetry as long as pulse data was of sufficient quality. We used Poincare plots to visualize the autocorrelation structure of heart rhythms. Each heartbeat interval is plotted as a function of the next interval. This Poincare plot was derived from a single participant's ECG measurements in a previous study, while this one is from our analysis of a HCP participants using pulse oximetry data. The diagonal patterns both indicate a steady heart rhythm, as each interval is similar length as the next one. Arrhythmias are often characterized by abrupt changes between successive intervals, producing unique off-diagonal patterns. Here is a triple lobe pattern defined using ECG in a patient by Esper and colleagues, which they found to be associated with the presence of multiple PVCs. And here's our replication of that same pattern using pulse oximetry data from a HCP participant. Notice how the abrupt changes in interval lengths map onto the different off-diagonal islands on the plot. For example, the purple intervals are produced by a short interval followed by a long interval. Looking through the entire trove of HCP physiological data, we identified many resting state fMRI runs with triple lobe point correlate patterns. Here are plots of 8 runs taken from 8 different participants with the same triple lobe pattern. These patterns became even more clear when computed from a shorter segment of data due to the reduction of baseline drift in heart rate. Inspection of the pulse time series confirmed the presence of ectopic beats and the resulting short-long interval patterns associated with PVCs. We found that PVCs were even more recognizable when plotting continuous heart rate, which revealed salient spike dip patterns. Continuous heart rate was calculated simply from the pulse beat intervals. We identified at least one PVC in about 5% of all HCP resting state fMRI runs that had pulse oximetry data of sufficient quality. In order to better characterize their bold signal correlates, we passed through all PVCs a series of stringent exclusion criteria. PVCs that appeared too close to each other or to the start or end of scans were excluded. This sometimes meant that a participant with lots of PVCs that were clustered ended up contributing fewer PVCs than one that had fewer PVCs but which were more evenly spaced. We also excluded PVCs with high levels of respiratory fluctuations or head motion, thus ending up with a total of 66 PVCs across 14 participants for our analyses. Let's look at a segment of a single resting state fMRI run which was flagged as containing PVCs in the pulse oximetry data. On the top, we have continuous heart rate in red. We have 2 measures of head motion, frame-wise displacement and DVARs. We have the raw respiratory trace in blue and respiratory variation, which is derived from the respiratory trace. At the bottom, we have the global fMRI signal in black and a carpet plot where each row shows activity in a different location in gray matter. If we look at the heart rate trace at the top, we can easily identify 4 events. Zooming in on the original pulse waves, we can see that 3 of the 4 events correspond to PVCs and 1 to normal rhythm with a gradual change in heart rate. We can see that the heart rate fluctuations with normal rhythm coincide with a deep breath on the respiratory trace. This is accompanied by head motion. The fMRI signal shows an initial spike followed by an extended drop. This pattern is consistent with previous work by Rasmus Bern and others. When we look at the PVCs, we also see a global fluctuation in the fMRI signal, except it's going in the opposite direction. First a drop, and then a spike. Importantly, in 2 of the PVCs, there are no accompanying head motion or respiratory fluctuations, as we see in deep breaths. In the 3rd PVC, we do see a pause in the respiratory trace and some associated head motion. This matches the general clinical observation that PVCs in some cases are accompanied by palpitations that cause discomfort. This could explain abrupt changes in breathing or changes in posture. However, in most cases, PVCs are silent events that are only detected incidentally during ECG recordings. Interestingly, the PVC with corresponding head motion and respiratory pause produced the same overall fMRI response pattern as the other 2 more silent PVCs. Let's look at an additional 5 PVCs, each from a different HCP participant. In each example, the PVCs are associated with the same pattern of a drop followed by an overshoot in global bold signal. Importantly, there are no large accompanying fluctuations in respiration or head motion measures. Looking at the mean heart rate and mean global fMRI signal responses, 2 things are readily noticeable. First, the drop in heart rate almost perfectly coincides with the drop in fMRI signal. The response minima at 1.8 seconds is much too fast to be due to neurovascular coupling and most likely reflects a drop in cerebral blood flow. Second, the overshoot's peak is substantially delayed in the fMRI signal at 5.7 seconds compared with the peak of the heart rate response at 3.6 seconds. This delay is interesting and may be explained by a combination of vascular and neurovascular mechanisms. The comparison of the mean response profiles of PVCs and deep breaths support the earlier observations in individual examples. PVCs produce similar magnitude changes in fMRI signal as deep breaths, but with much faster dynamics and without large respiratory fluctuations or head motion. Another striking feature of PVCs was the high consistency of the bolt signal response. Here are 7 PVCs from a participant, and here are 9 from another participant. Notice the strong correspondence within and between the participants. The overall pattern of a dip and overshoot is also visible across all 66 PVCs. However, we do see some individual variation in the amplitude of the responses. We hypothesize that if these effects are primarily blood flow effects, then they may be explained by individual differences in the pulse wave features. We first examined the impact of the length of the post-ectopic intervals. We hypothesized that longer post-ectopic intervals would lead to larger drops in cerebral blood flow and thus lower global fMRI signal minima. Indeed, we found a strong correlation between post-ectopic interval length and bolt signal minima. The colors in the 2 plots refer to corresponding bins denoting different post-ectopic interval lengths. A mere 20% increase in post-ectopic interval lengths generated substantially greater drops in bolt signal. Next, we examined the impact of the amplitude of ectopic pulses. We predicted that a smaller pulse would produce a bigger drop in bolt signal, as it may suggest lower levels of perfusion. Indeed, we found a strong correlation between ectopic pulse amplitude and bolt signal minima. Again, the colors in the 2 plots refer to corresponding bins denoting different ectopic pulse amplitudes. When we entered the 2 measures into a single GLM, it explained 66% of the variance in bolt signal minima. This provides strong evidence that individual differences in the magnitude of bolt signal responses to PVCs are closely related to their pulse wave properties. In summary, Poincaré plots offer a promising approach to identify PVCs during resting state fMRI scans. PVCs elicit large-scale global bolt signal fluctuations with a highly conserved temporal profile between and within individuals. PVCs can produce large-scale bolt signal fluctuations in the absence of respiratory fluctuations or head motion. And finally, pulse oximetry features such as post-ectopic interval length and ectopic pulse amplitude explain individual variation in global signal response amplitudes following PVCs. I would like to thank the following 5 people for their contributions to this project, with special thanks to my supervisor, Thomas Yo, and Jonathan Power for their mentorship.